Today we are going towards embryogenesis. Embryogenesis means the development of embryo. Genesis means to synthesize, to develop. To develop the embryo is called the embryogenesis. So the zygote, whenever it converts into the embryo, is called the embryogenesis. You know that how that zygote is produced? When the egg cell, whenever it fuses with the one of the male gamete, then it converts into a diploid zygote, and whenever it converts into a embryo, then it is called the embryogenesis. So after completion of embryo, the or after completion of endosperm, the zygote is produced, and embryo synthesis starts. You know that here during fertilization, what happens here? This is egg cell. This is the egg cell, and when egg cell, it fertilizes or it fuses with one of the male gamete. This is the male gamete, and another male gamete, whenever it fuses with the secondary nucleus, then secondary nucleus is converted into primary endosperm nucleus, and primary endosperm nucleus is converted into the endosperm. So we have seen there are the different types of the endosperm on the basis of development. But when this zygote, diploid zygote, whenever it fuses with the one of that male gamete, one of that this is the male gamete, one of that male gamete, here it is zygote is produced, and when the zygote, whenever it converted into the embryo, this process is called the embryogenesis. Suppose this is the zygote, diploid zygote. This diploid zygote, when it divides mitotically, then it produces the two cells. This is the lower cell, which is toward that micropylar end, and this cell is called the suspensor initial. It is a larger one, and the cell which is toward the chalazal end, this is the cell which is toward that chalazal end, this cell is called the embryonal initial. This embryonal initial cell, it is toward the chalazal end, and the micropylar, toward the micropylar, this cell is a larger one. This cell is the larger one, and it is toward the micropylar end, and this cell is called the suspensor initial. Then here that suspensor initial, it divides mitotically and produces the 6 to 10 cells, and this is called the suspensor. The cell which is toward the micropylar end, it becomes swollen and it acts as a haustoria, and the cell which is lower to that embryonal cell is called the hypophysis. This is called the hypophysis. Then due to that suspensor cell, so the 6 to 8 cells are produced, and this due to that 6 to 8 cells, here this suspensor cell, it push the embryo into that endosperm. You know that the endosperm, here this is the endosperm, these endosperms provide the food material for the development of that embryo. So it has to push into that endosperm, and that's why here the suspensor cells are produced. Then the embryonal cell, it divides 3 successive mitotic divisions. Here 3 successive mitotic division takes place. How many? 3 successive mitotic division takes place, and octant is produced. Here the octant, that is the 8 cells are produced. So the by 3 successive mitotic division, octant is produced. Then out of that 8 cell, the lower 4 cells or the lower tiers, they are converted into the hypocotyle and radical. They are converted into the hypocotyle and radical, and the upper cells, they are converted into the plumule and cotyledons. Again here the cell divides and redivides to produce the heart shaped structure, and it gives rise the terminal plumule and two lateral cotyledons. Then the cell which is it is called the hypophysis. Whenever we call it is a hypophysis, this hypophysis, it convert into the radical and root cap. Again it divides and redivides to produce horseshoe shaped structure, and it gives rise to the plumule and the cotyledons. As like this, here the embryo is synthesized. So the suspensor cell, what is the function of suspensor cell? It push the embryo into the endosperm, and the embryonal cell, it divides mitotically to produce octant. Again it divides and redivides to produce heart shaped structure. Again it divides and redivides to produce the horseshoe shaped structure. Okay. uh, then here the development of embryo is the similar in dicot as well as in the monocot up to the octant stage. Look at here, up to that octant stage, here this the division here up to that octant stage. Here look at here, up to that octant stage, here the division or the development of the embryo is the similar in monocot as well as in a dicot. But here in the after the octant formation, the single cotyledon is produced, and the single cotyledon is called the scutellum. As well as here the protective coating of that plumule is called the coleoptile. Protective coating of this radical is called the coleorhiza. So the difference takes place after the octant. So in monocot, the single cotyledon is at a terminal position, plumule is at a lateral position, single cotyledon is called the scutellum, protective sheath is called the coleoptile, and protective sheath of radical is called the coleorhiza. Then here the ovule is converted into the seed, and ovary is converted into the fruit. As like this, here the embryogenesis and the endosperm synthesis, we have seen that. So the primary endosperm nucleus, it convert into the endosperm, and zygote, it convert into the embryo. And embryo, how that embryo is produced or how that embryo is synthesized, we have seen. The plumule, cotyledons, they are produced from that embryo. From plumule, here the shoot system is developed, as well as from radical, here the root system is developed. Okay, so we have seen here, we have completed the embryogenesis.